Now we are going to move to the next topic, that is inflammatory mediator. Another important topic for concept as well as exam. Now we are going to study about inflammatory mediators. So what are the inflammatory mediators? How we can divide inflammatory mediators? We can divide inflammatory mediators into two broader groups. Either it can be a cellular mediator, or it can be a plasma mediator. Cellular mediator or plasma mediators, right? Cellular means within the cells they will be present like a mast cell or neutrophil. Plasma mediators are synthesized by liver, right? Plasma is synthesized by liver and they will be inactive. During inflammation they will be released and then they will become activated. So that is the difference between cellular and plasma. Plasma, what are the plasma inflammatory mediators? Plasma inflammatory mediators, example I will give you, bradykinin, and second example I will give you of complements, right? So complement and bradykinin, these are two important examples of the plasma mediators which we will discuss later on. Come back to the cellular mediators. What are the cellular mediators? So again we will divide cellular mediators into two broader categories. One is the preformed cellular mediator and second will be newly synthesized cellular mediator. So there are two groups. Now you can see there are two groups. One is called preformed. Preformed means, as you can see, the name itself is telling, which is already present within the cell. Already present within the cell, preformed mediator. And second group is the newly synthesized. Second group is the newly synthesized, which is synthesized during inflammation. So they are not already present. They are not already present. When inflammation is going on, that time they are synthesized, newly synthesized inflammatory mediators, right? So preformed mediator and newly synthesized mediator. What are the preformed mediators? We will discuss in brief, right? Preformed mediators. We can divide preformed mediators, or there are four important groups in preformed mediators, right? So I'm writing preformed mediators, four important groups, right? Number one is histamine. Number two is serotonin. Number three is heparin. So heparin, right? And number four is lysosomal granules, which is present inside the neutrophil, right? So lysosomal granules, these are the four important preformed mediators, right? This was the question also in DNB and All India exam, which is not a preformed mediator: histamine, serotonin, heparin and cytokine. So cytokine is newly synthesized. During inflammation that will be synthesized, they are not already present. So that is why we have to remember histamine, serotonin and heparin, these are the already present, preformed. Where and how? Now we are going to discuss. Histamine, richest source is mast cell. Richest source is the mast cell. This is the another important MCQ which examiner asked, right? Richest source for histamine is the mast cell, and histamine is synthesized from histidine amino acid. They are synthesized from histidine amino acid. Another important question. So richest source is the mast cell, synthesized from the histidine amino acid. Serotonin, richest source is the platelet. And they are derived from 5-hydroxytryptamine, which was All India question in 2018. So they are derived from the 5-hydroxytryptamine. So they are also called as 5-hydroxytryptamine. 5-hydroxytryptamine is name of which mediator? That was the question. So 5-hydroxytryptamine is the name of serotonin. That was the question in All India exam 2018, right? Coming to the heparin. Heparin is another important example of preformed mediator which will be present inside the mast cell. So their richest source will be mast cells. So mast cells will be having heparin, right? Lysosomal granules which will be present in the neutrophil, we can see that they will be having three types of granule: primary, secondary and tertiary. So primary granule, they will be having secondary granule, and they will be having tertiary granule. So these are three types of the granules which we will see in the lysosomal granule of the neutrophil, right? Primary granule will be having myeloperoxidase. They will be having defensins, right? Cathelicidin. So these are the proteins which are present in the primary granule of the lysosomal granule. Secondary granules will be having lactoferrin and lysozyme. Right? So lactoferrin and lysozyme. Tertiary granule will be having gelatinase. So these are the lysosomal granules: primary, secondary and tertiary, which is present inside the neutrophil. So these are all example of the preformed mediators. So now you can see histamine, serotonin, heparin, lysosomal granule, all these are preformed mediators, right? So all these are preformed mediators, right? Newly synthesized means when they will be synthesized during inflammation, they are called as newly synthesized. So what are the components of the newly synthesized? Number one, here it is nitric oxide. Nitric oxide is not already present. They are synthesized during the inflammation, and they are synthesized from which amino acid? Nitric acid is synthesized from arginine. It is synthesized from arginine, and this was the question in this All India exam, right? This was the question in this All India exam. They are synthesized from arginine. So nitric oxide is synthesized from arginine. This was the question in this All India exam, right? So that is the point we have to keep in mind. Nitric oxide is synthesized from arginine. Second is platelet activating factor. So second thing which we are going to see is the platelet activating factors. So platelet activating factors. And third, as I have said, cytokines. And fourth, newly synthesized is arachidonic acid metabolite. Arachidonic acid metabolites, right? So arachidonic acid metabolites, they are usually derived from essential fatty acid known as linoleic acid. So that is another important MCQ which examiner asks. Arachidonic acid is derived from which amino acid? Linoleic acid, right? So these are the important points we have to keep in mind for the newly synthesized inflammatory mediator, right? So these are the newly synthesized: platelet activating factor, nitric oxide, cytokine, arachidonic acid metabolite. So we will discuss arachidonic acid metabolite and cytokine later on. So first we will discuss some of the salient features of these mediators. So as I said, we will discuss some of the salient features. So I am going to club those inflammatory mediator which are having common function at one place, right? So first I am going to club all these who are having common functions, like histamine, serotonin, platelet activating factor, and bradykinin, right? So all are having similar functions And what are those similar functions? I will write down first, right? So their functions, I'm writing about the functions of all these mediators, right? Histamine, serotonin, platelet activating factors, and bradykinin. So they are having similar function. What are those similar function? They are all involved in vasodilation, increasing vascular permeability, increasing vascular permeability, vasodilation, and smooth muscle contraction. Smooth muscle contraction or constriction. So smooth muscle constriction will be another important feature of this inflammatory mediator, right? So these are three important functions which will be common to all of them. So histamine, serotonin, platelet activating factor, bradykinin, all will be having this common function: vasodilation, vascular permeability increase, smooth muscle constriction. Smooth muscle constriction, in exam they will be writing with this name of the bronchoconstriction, right? So just be careful, both are same thing, bronchoconstriction. So examiner will be writing bronchoconstriction, so that will be the same thing as a smooth muscle. Because of the smooth muscle constriction only there is a bronchoconstriction, right? But apart from this, what I want to convey one message, ki histamine, serotonin, platelet activating factor. See what I am saying? Histamine, serotonin, and platelet activating factor, they can cause vasoconstriction also. So vasoconstriction and vasodilation both will be caused by histamine, serotonin, and platelet activating factor. Bradykinin is one important all India question. Okay, what is the most important function of the bradykinin? See the language of the question, most important function of the bradykinin. Most important function, when language is like this, most important function of the bradykinin, your answer should be pain. That's all. Most important function for the bradykinin will be pain, and that was the answer in all India exam. Most important function is the pain. This is the one language. But now look at the second language which examiner had used in all India exam. Most important function of the bradykinin in acute inflammation. In acute inflammation, see the wordings has been changed. In acute inflammation, what will be most important function? Now you say, what will be most important function of the bradykinin in acute inflammation? So in acute inflammation, hallmark is the most important function. And what is the hallmark of acute inflammation? Increasing vascular permeability. So increasing vascular permeability. So increasing vascular permeability will be the most important function of the bradykinin in acute inflammation. So these are the two questions which examiner asked based on the language of the MCQ. You have to decide the right answer. Most important function, answer should be pain. Most important function in acute inflammation, answer should be increased vascular permeability, right? So this is how we can understand these four common things at one place. Now I am moving to the next topic. Next topic in this inflammatory mediator I want to discuss is the arachidonic acid metabolites, right? So next topic which I am going to discuss here is the arachidonic acid metabolites. In short, I will write AA, arachidonic acid metabolites and their pathway. As I have told you, arachidonic acid, they are derived from the phospholipid. Phospholipid, which is containing essential fatty acid, linoleic acid. Already I have told you linoleic acid. So that is important MCQ, like linoleic acid, essential fatty acid, right? They will be containing essential fatty acid, linoleic acid. So this phospholipid, which is containing linoleic acid, will be broken down into the arachidonic acid metabolites. They will be broken down into arachidonic acid metabolites, right? By which enzyme? So now you can see the name itself is telling phospholipase. So phospholipase A2 This enzyme will break down the phospholipid into arachidonic acid metabolites, right? And now after this phospholipase action, when phospholipid has been broken down into arachidonic acid metabolites, they will follow two pathways. Now this arachidonic acid will be following two pathways. One is called as lipooxygenase pathway. One is called as LOX pathway. LOX means lipooxygenase pathway. Lipooxygenase pathway. So this is the number one, lipooxygenase pathway, and second will be COX, cyclooxygenase pathway. Cyclooxygenase pathway. So we are seeing arachidonic acid metabolism. They will follow two pathways, lipooxygenase and cyclooxygenase. So we are going to discuss these two pathways. In lipooxygenase pathway, you will notice there are two groups of enzymes, right? One is called 5-lipooxygenase enzyme, and second group is containing 12-lipooxygenase enzyme, right? So these are the two groups, 12-lipooxygenase, 5-lipooxygenase. So 5-lipooxygenase pathway, they will be forming leukotriene A4. So in this leukotriene A4 will be formed, and from leukotriene A4, you will see they are getting converted into leukotriene B4, leukotriene B4, leukotriene B4, leukotriene C4, D4 and E4. So 5-lipooxygenase pathway, we can see leukotrienes are formed. Lipooxygenase pathway, leukotrienes are formed. Leukotriene B4 is chemotactic to the neutrophil. So that is the important MCQ, chemotactic for neutrophil. So that is the important MCQ, chemotactic for neutrophil is the leukotriene B4. Whereas leukotriene C4, D4 and E4, these are collectively known as slow reactive substances of anaphylaxis. So they are collectively called as slow reacting substances of anaphylaxis. This is important abbreviation which examiner asks in exams, right? So SRSA stands for slow reacting substance of anaphylaxis. So slow reacting substances of anaphylaxis, this is the SRSA, right? Slow reacting substances of anaphylaxis, leukotriene C4, D4 and E4, right? So this is about the leukotrienes. Now coming to the see what I have shown you, these are all inflammatory which will cause inflammation. On this basis, there was a question in AIIMS exam which is anti-inflammatory. Which inflammatory mediator is anti-inflammatory? So remember, when 12-lipooxygenase pathway will be followed, then they will be forming lipoxin. Then they will be forming lipoxin. And remember, lipoxins are anti-inflammatory in action. So that is the important answer. Lipoxin is anti-inflammatory, right? So lipoxin is anti-inflammatory. This was the question in older AIIMS exam, right? So anti-inflammatory is the lipoxin. So why it is anti-inflammatory? What is the inflammation? Inflammation, you can see leukotriene inflammatory. They are bringing neutrophil, means they are attracting inflammatory cell like neutrophil, right? So same way, lipoxin will be opposing the entry of the neutrophil. Mean to say that they are the endogenous negative regulator of neutrophil or leukotriene. So lipoxins are also known as endogenous, because they are formed within our body, endogenous negative regulator of leukotrienes. So negative regulator of the leukotrienes. So these are the negative regulators of the leukotriene, and as I said, leukotriene they will be involved in chemotaxis of the neutrophil. So what will happen here? Because of this, what should be the function? Now by seeing this, you can understand what should be the function of lipoxin, which was the question in all India exam. So they will inhibit So lipoxin will inhibit neutrophil chemotaxis. They will inhibit neutrophil chemotaxis. So this will inhibit neutrophil chemotaxis, number one, and number two, to prevent neutrophil to come to the site of inflammation. What else they can do? So this lipoxin will activate. See, this lipoxin will activate macrophage for phagocytosis of neutrophil, right? So now we can see that it is for macrophage activation for phagocytosis of neutrophil. So this will be the right answer. So now you can see this was the question in All India 2016. Okay, what is the function of lipoxin? See, earlier AIMS question was which is anti-inflammatory. Lipoxin is anti-inflammatory. Now the question is what is the function of lipoxin? So try to understand. Topic will be same and question will be different. So this was question. Okay, what is the function of lipoxin? So option number 1, lipoxin inhibits neutrophil chemotaxis and activate macrophage for the phagocytosis of the neutrophil. That is how they behave anti-inflammatory. So lipoxin is anti-inflammatory mediator, right? So this is about lipoxygenase pathway. When we are seeing cyclooxygenase pathway, in cyclooxygenase pathways, arachidonic acid will be converted into prostaglandin G2. Then prostaglandin G2 will be forming prostaglandin H2, right? So now we can see the prostaglandin G2, prostaglandin H2 will be formed, and these two are common substrates. Right? So PGG2 and PGH2 is a common substrate for prostacyclin PGI2. PGI2, we all know that this is called as prostacyclin. Right, so number 1, prostacyclin, number 2, thromboxane A2, and number 3 is the prostaglandin. So these are the 3 things they will be forming, right? Prostacyclin, thromboxane and prostaglandin will be formed by the common substrate PGG2 and PGH2, right? So now we will see what is prostacyclin and thromboxane A2. Prostacyclin, if you look at the origin, right, what is the richest source of prostacyclin? This was all India question. Endothelium, right? Endothelium is the richest source of the prostacyclin. Endothelium is the richest source of the prostacyclin. That is the all India question, right? What is the richest source of the thromboxane A2? This was the PGI question previous year, right? So it is platelet. Platelet is the richest source of thromboxane A2, right? So when thromboxane A2 is there, what will be the function of thromboxane A2? What will be the function of prostacyclin? We can easily make it out, right? Why I am saying we can easily make it out? Because both are antagonists to each other, means both will be opposite to each other. Thromboxane, whatever function thromboxane will do, exactly opposite function will be of prostacyclin. So let us see thromboxane A2 first. So thromboxane A2, what is the function? What is the function of thromboxane A2? As you can see, the name itself is telling thromboxane, platelet. So they will be involved in platelet aggregation. So platelet aggregation will be promoted by, so these are platelet aggregators. So we can say that thromboxane A2 is a platelet aggregator. And whenever you see platelet aggregator, they will release the enzyme present in their granule, which will promote vasoconstriction. So whenever there is a platelet aggregation, there will be vasoconstriction. Whenever there is a platelet aggregation, there will be vasoconstriction. So because of the platelet aggregation, there will be vasoconstriction. So platelet aggregation will cause vasoconstriction. Whereas opposite function, now you can see prostacyclin will be having opposite function. So what could be opposite function of the prostacyclin? So prostacyclin will be associated with anti-aggregators, platelet anti-aggregators. So these are the anti-aggregators of the platelet. And if they are anti-aggregator of the platelet, what they are going to do? Vasoconstriction or vasodilation? Answer is vasodilation. So vasodilation and anti-aggregators are the prostacyclins. So these are the two important things which examiner had asked in previous year PGI exam also. So just keep that in your mind. This topic, entire topic is very famous topic, very often asked question in PGI as well as All India exam. Come to the prostaglandin, right? This All India, previous year All India and PGI exam. All exam they had asked this question, right? So now I'm going to tell you about prostaglandin. So here I will tell you three types of the prostaglandins which I wanted to discuss. One is the prostaglandin E2 alpha and second is the prostaglandin D2 alpha and prostaglandin F2 alpha, right? So these are the three prostaglandins. PGE2 alpha was All India question previous year and PGI exam. So prostaglandin E2 alpha is associated with fever and pain. So that is important. Fever and pain. And that was the question in All India exam. Fever and pain, they are associated with prostaglandin E2 alpha. So this was the question. Prostaglandin E2 alpha is associated with fever and pain. Whereas prostaglandin F2 alpha and D2 alpha, they are associated with neutrophilic chemotaxis. If you remember, just now we have discussed in chemotaxis, right? So neutrophilic chemotaxis is mediated by PG2 alpha and PGF2 alpha. So PGD2 alpha and F2 alpha is associated with neutrophilic chemotaxis. So these are the important points we have to keep in mind while studying about the prostaglandins or arachidonic acid metabolites. Now we are going to see next topic is a very important topic for exam, that is cytokine. So next topic we are going to discuss will be cytokine.